Greetings and welcome to the United States Transhumanist Party Virtual Enlightenment Salon. My name is Gennady Stolyarov II, and I am the chairman of the U.S. Transhumanist Party. Here we hold conversations with some of the world's leading thinkers in longevity, science, technology, philosophy, and politics. Like the philosophers of the Age of Enlightenment, we aim to connect every field of human endeavor and arrive at new insights to achieve longer lives, greater rationality, and the progress of our civilization. Greetings, ladies and gentlemen, and welcome to our U.S. Transhumanist Party Virtual Enlightenment Salon of August 6th, 2023. Today we have a fascinating conversation in store for you about the state of longevity science, particularly in the years since the publication of this book, Ending Aging, co-authored by Dr. Aubrey de Grey and Michael Rae. So we will discuss how longevity science has advanced since 2008 when this book was published. And joining us is our distinguished panel of U.S. Transhumanist Party officers, members, and advisors, including our Director of Visual Arts, Art Ramon Garcia, our Legislative Director, Jason Geringer, our Director of Longevity Outreach, Ben Ballweg, our Foreign Ambassador in Spain and Technology Advisor, Dr. Jose Cordero, our Biotechnology Advisor, Founder and CEO of Sierra Sciences, Dr. Bill Andrews, and David Wood, who is the founder of London Futurists. He's also executive director of LEVF, the Longevity Escape Velocity Foundation. And our special guest for today is Michael Rae, the co-author of Ending Aging, the rejuvenation breakthroughs that could reverse human aging in our lifetime. He is also the author of 5 scientific articles and commentaries in peer-reviewed scientific journals. Much of his work has been devoted to elucidating the SENS platform for anti-aging biomedicine for a popular audience. SENS stands for Strategies for Engineered Negligible Senescence. His undergraduate minor was in biology. He is a longtime member and one-time board member of the Calorie Restriction Society, a main contributor to the Society's how-to guide, and was the core scientific investigator with the CR Society Cohort Study. He currently works as the science writer for the SENS Research Foundation, and you can read a lot of his articles on the Sensible blog. I would encourage you to check that out. And now, Michael, thank you very much for joining us, and the floor is yours.
Good day, Gennady, and to everybody. We wrote the book back in 2006. It was published in 2008. And I was really delighted to accept your invitation to present on the progress that we have made in the last 15 years, because progress we definitely have made. And it was actually really encouraging to me to put this together, because it meant that I had to sort of review, wow, we really have made a lot of progress in the last 15 years. So I guess we can get on with it. Both for people who are not familiar with SENS and as a refresher for people who at least loosely are, I'm going to start by just reviewing the basics of the SENS approach itself, which is a very specific way of strategically tackling the ending of human aging. So aging is a result of a lifelong process. I'm guessing people can't see my pointer, can they?
No, we can only see the slides.
That's unfortunate. Okay, so I can't point at things. That will be limiting, but not a disaster. Aging is a result of a lifelong process. So the processes that keep you alive, all the biochemistry in your cells, that's what we mean when we talk about metabolism. So not just the metabolic rate, how fast you're burning calories, but all the cellular processes, all the biochemistry that keeps the organism living. But because those are just physical and chemical processes, they lead to molecular and cellular byproducts that were not intended that can accumulate in your cells and tissues as damage. Um, and because form follows function in biology, when you lose the structure of a biological unit, that unit is no longer able to fulfill its function. And over time, the progressive accumulation of damaged units means that you lose the function of that organ or that tissue. And when enough of that damage accumulates in a given organ or tissue, that's when you get what we categorize as diseases of aging. So is this lifelong flow from metabolism to damage to pathology that causes and constitutes aging. So what are you going to do about that? The approach that is sort of standard of care medicine today is what Dr. de Grey used to call geriatrics. So this is the idea you already have some pathology of aging that has accumulated in your body. So you have heart failure, you've had a heart attack, you have diabetes. What are we going to do to prevent that from acutely killing you? So you have heart failure, for instance. Your heart can no longer eject blood properly as it should. Well, we are going to give you one drug that is going to regulate the rate at which your heart beats so that it doesn't kill you. And we are going to give you another drug to regulate fluid in your body because that is a byproduct of the way your heart is trying to deal with the fact that your heart is not pumping properly. This does nothing to the underlying pathology. It simply keeps you from acutely dying of it. And of course, as you are, living your life with these drugs in your system, that damage is continuing to accumulate both in your heart and elsewhere. And so you just become sicker and sicker, and eventually either that disease or something else kills you. So hooray that we have that option, but not really the best thing we want to have happening. The second kind of approach is sort of the best of today's preventative medicine, plus what we today call geroscience, what Dr. de Grey used to call uh, gerontology. So here the idea is let's go back to the beginning here. What are the cellular molecular processes that drive the accumulation of aging in the body? Can we modulate those processes in a way that will cause you to accumulate less damage and therefore not develop the pathologies of aging as quickly? So this is everything from getting your ApoB and your blood glucose under control to drugs like rapamycin or canagliflozin or calorie restriction or inhibiting the IGF-1 pathway. All these things, the strategy is basically let's damp down on the processes that are causing this damage to accumulate so that we can delay the onset of disease. There's a couple of problems with that. One of which is that although you can slow down the rate at which damage accumulates by those kinds of modulations, you can't stop them because you can't stop metabolism except by death, which will rather defeat the purpose. Um, and the other problem is that, by the time you start applying this approach, a person has already accumulated a significant amount of damage. If you start at the age of 40, you haven't done anything to deal with all the aging damage you've accumulated for the previous 40 years of your life. And even if we started a person at age 20, the same would apply. You are merely, as it were, delaying the inevitable. So the insight that Dr. de Grey had in 2000 was there is a third approach that no one is really being considering, which is directly attack the damage itself. And this is a very, very powerful new approach. And the reason for that is, on the one hand, you are preventing the pathology from happening by undoing the damage that will lead to pathology, but you are allowing metabolism to operate normally. If you take a person who has 200 milligrams per deciliter of blood sugar in their body and bring it down to 100 milligrams of blood sugar in their body, you are reducing the rate at which glycation happens and thereby delaying the complications of diabetes and doing, essentially nothing but good. But if you then try to bring that down from 100 to 70 in order to further slow down the rate at which that damage occurs, you are going to be in danger at any particular time of slipping into a hypoglycemic shock. There is just there's a limited amount of things you can do by interfering with metabolic processes, whereas damage by its very nature is aberrant uh, accretions onto the structures of life. There is nothing but good from removing damage, and therefore you can remove that damage without causing these dangerous side effects that metabolism interference can do. And you can, if you do it comprehensively, indefinitely forestall the pathology of aging. And as part of that, uh, Dr. de Grey developed this schema, the 7 categories of damage, whereby he has divided all the various different kinds of cellular and molecular damage that accumulate in the aging body and cataloged different prototype methods whereby we could remove, repair, or replace all those damaged functional units. And when you do that comprehensively, systematically, you could thereby indefinitely postpone uh, aging-related pathology. So this is just to illustrate what we've been talking about so far. If you take a person who is, say, 40 years old, and you half the rate at which damage accumulates in middle age, you have forestalled the falling across of the threshold of frailty, but you have only somewhat delayed that by a very modest degree. Whereas if you instead cut away half the damage, repair half the damage, you have a much, much larger effect in terms of both total health span and remaining health span from the time you first apply therapy. And the advantage is that it's iterable. So if I come back again and I further cut the amount of damage in the body by half again, I have actually made you better off than you were even before you started receiving therapy. And you have been better than the previous round you first applied therapy. Whereas doing the equivalent on the slowing side doesn't have anything close like that effect and can't be repeated over and over again because of the limitations that the body needs to proceed with its metabolic processes just in order to keep you alive. So, in terms of what has happened since we laid out that strategy in anti-aging, and indeed, since Dr. de Grey first came up with that strategy back in the year 2001. Um, this is sort of an ironic beginning. This is not SENS at all. This is old school gerontology, but this was a really important breakthrough in 2009. It's kind of hard to think back to the world we were in back at that point. Uh, in 2000, in 2006, in 2008, in the early of the parts of the year, uh, we had 0 drugs that could retard aging in any mammal. Certainly, there were many people who were making claims about this drug or this dietary supplement or that, but there was nothing convincing in terms of a molecule that I could give to a person that would retard aging in that person. Um, there were mutations and there was calorie restriction, and that was it. In 2009, for the first time, we had a real medical type intervention that, at least in mice, could retard aging. And of course, now we know that it does the same thing in a variety of different species. It looks like it's doing it in dogs. We'll know more about that in a couple of years. And we know that it does this very robustly and not just in a one-off experiment. So that played a really important part in changing the minds of both the public and the scientific community in terms of, yeah, aging is actually something tractable, which was kind of obvious from everything we knew since 1935, but it was not yet demonstrated that you could do that in a realistic intervention. And this really changed people's mindsets. And scientists started saying the unspeakable about this, which is, we could actually do something about aging in the real world. So down here in the bottom half of the screen, you are seeing the appearance in the biomedical literature of, on the left, either the term rejuvenate or rejuvenation, and on the right, the combination of aging and some variation of intervention. And you can see that there were very, very few occurrences of either of these formulations of the problem up right until about 2009, 2010. And suddenly, this started appearing rapidly and at an ever increasing pace year after year thereafter in the biomedical literature. So this really changed the mindset of the scientific community. Um, Above that, you're seeing two screenshots. One is an archive screenshot from 2009. One is just from a couple of days ago for the Bucks Institute, which is a perfectly respectable scientific organization working on aging. And back in 2009, they were cautiously saying, although this was more than a lot of other organizations were willing to say, our mission is extending health span, the healthy years of life. Whereas now they are saying somewhat more boldly, live longer, better. And I really started to think that we were changing the mindset of the scientific community back in the early days of the COVID-19 pandemic. I was listening on the radio in May of 2020, and they put on a underwriting spot on the local public radio program, part of which was, we believe an extra 20 years of life is possible. And that was something that, nobody aside from Dr. de Grey and a few other, considered fairly radical people at the time would have been willing to say. And suddenly that was sayable. Um, and similarly, there was a real change in the way the press started covering the issue and the contact that SENS Research Foundation was getting from the public in terms of people actually taking seriously the idea that we could do something with this and that it was a good idea to do so. Another important change, the idea of a divide and conquer approach, has really become a very mainstream sort of approach. So people who follow aging research will know these are the so-called hallmarks of aging. This is a different schema, and I would argue a less useful schema than the SENS 7 that Dr. de Grey devised, but it has really become a mainstream idea that we don't only have to think of aging in terms of one thing that is going to turn down every single thing that is going wrong in an aging body, that we can categorize aging into a variety of different changes and go after them individually, and that by doing that collectively, we can also very effectively intervene in aging. We can come back to the reasons why I think that the hallmarks is not really the most effective approach to doing that compared to the SENS 7. But the point is that it's become a very mainstream idea in the aging community that this divide and conquer sort of approach can do the job. Another thing that has happened that is critical to our eventual conquest of aging is that we now actually have a rejuvenation biotech industry or a longevity biotech industry. This was something that Mike Cope, our former CEO at SENS Research Foundation, really wanted to emphasize and really did a lot of effective work on. In particular, the Rejuvenation Biotechnology 2014 conference, where the explicit goal was to build a rejuvenation biotechnology industry where we now really have an ecosystem whereby venture capital and scientists who are actually working on intervention in aging as opposed to aging as a biological curiosity, um, people who are working on what are in fact rejuvenation biotechnologies who don't even realize that they are working in aging, uh, people back then who were working in aging but never spinning out companies. We had a serious problem with lack of regulatory reform. All of those things are very rapidly changed. And right now, today, we have a whole ecosystem that has sprung up whereby all of those pieces are increasingly in place. This is just a small number of companies selected by Stadia, representative of the fact that we have a broad variety of startup companies working in aging that are tackling all kinds of important things towards eventual conquest of aging. That includes many spinouts that have been formed either directly from research that SENS Research Foundation has conducted in our own labs, or research that we have that we have sponsored in external labs like Yale University for, uh, do I not have it here? Gosh, I don't. For Revel, oh yes, I do. For Revel up here, or independent scientific organizations like Oshin. Um, many companies out there getting funded. The regular biotech press is covering these. This is a whole new universe we are operating in. So let's go through category by category some of these standard examples of how we have made progress against all the different categories of aging that we'll need to eventually conquer aging. This is quite new. So we now have uh, essentially three. The last of them hasn't actually gotten approval yet, but I don't think anybody has any doubts that it will. We now have three therapies that directly remove different forms of beta amyloid from the brain that have all been proven to slow down the rate of degeneration of cognition and function in people who are in the early stages of Alzheimer's disease. There are people in the aging community who are downplaying, I think for unreasonable reasons, what a breakthrough this is. In the entire history of humanity, we have never had any kind of intervention that has actually slowed down the rate at which people degenerate into Alzheimer's disease, dementia, and death. And for the first time, we have therapies that will actually slow that process down by directly removing the damage in the aging brain. Um, and in particular, if you look at people who have less of the downstream damage in the form of tau in their brains, we're talking about a slowdown of as much as 40%. I'm going to skip through a couple of slides because the time is going to run away from me if I don't. But we can come back to this, why we have not done even better on this and how we could do even better in the future by being more comprehensive and more early. Um, an important opportunity to prove that we can do that is open to anyone out there who is age 55 or older. So donanemab, the rejuvenation biotechnology I was just talking about in that previous slide, uh, the company responsible for that, Eli Lilly, in combination with the National Institutes of Health, have now launched the AHEAD study. They are taking people with no evidence of cognitive impairment, and they are giving them donanemab with the intention of showing that they can prevent the downstream damage from accumulating and thereby eventually potentially prevent Alzheimer's disease from developing in the first place. And a second rejuvenation biotechnology has just been published in the last month. And this is removing a different kind of extracellular aggregate, a different amyloid, this time from the heart. This is transthyretin, TTR, amyloid in the heart. These are antibodies that directly remove that from the heart tissue. And you can see that here in the slides. These are two people on the left that you can see progressively over the course of the trial had their amyloid actually removed from their hearts. On the right, you're seeing the extracellular material, which essentially is amyloid, similarly removed off of that. And I don't have it here in the slide, but they showed both that there was less damage to the heart as a result, and that people had functional improvements, such as improvements in the 6-minute walking test. SENS Research Foundation, some years back, sponsored research in this area that has not yet been turned into a working therapy for humans, that instead of trying to pull out the amyloid from the heart, would actually chop it up. So these are things that are called catabodies. These are antibodies that don't merely bind to their targets, but actually cleave them into smaller units and eventually allow the body to hydrolyze them. And this was very powerful, at least in the lab tests that we've been able to do to date on it. Their current generation, which is better than the one that was published in this paper, removed very high levels of this material and cleaved it into tiny, tiny, tiny little particles sizes. So another area is apoptosens. These are abnormal cells that accumulate over time in our bodies and contribute to age-related pathology. Of course, the most famous of these is so-called senescent cells. These are cells that develop as a tumor prevention strategy in the body. So when a cell has undergone abnormal changes that will allow it to become a cancer, there is a program in the cell that will turn off its ability to replicate itself. And that prevents cancer from forming. But unfortunately, if these cells are not rapidly removed, and they aren't always, they can cause other problems in the body because, amongst other things, of secretions that they issue out that cause inflammation and that degrade the local tissue and that can cause other problems in aging and are responsible for a large part of the pathology of aging, as is clear from the fact that when you remove these cells from tissues, not only do the function of the local cells improve around it, but the organism actually lives longer. Excuse me, they don't live longer, but they do become healthier. They have a variety of age-related pathologies, either delayed or, in some cases, reversed. And an important development in the last 15 years or so has been that we now have actual small molecules that will do this. These are so-called senolytic drugs that destroy senescent cells in the body and that, again, prevent or slow down or in some cases reverse age-related pathology in mice. And we now have multiple human trials testing this idea. We've had a couple of failures in this space, I have to say, as well. So this has not been a complete rah-rah success. But for instance, Unity has recently had a very successful phase 2 in diabetic macular edema, which is a disease of the eye that people with diabetes get that is closely associated with senescent cells. So this is a really important area in which SENS has made progress. Um, One of the ways that the so-called SASP, these secretions that are released by senescent cells, causes further pathology in the body, which has more recently been discovered, is this phenomenon of secondary senescence. So this is the idea that you have a cell that has undergone senescence because of DNA damage or because it is replicated too many times or for another reason, it is releasing these SASP factors. Those SASP factors can actually cause either a neighboring cell or in some cases cells that are very far removed from it to become senescent. And so you get this domino effect where one cell drives a second cell senescent, drives a third cell senescent, and so on and so forth. So you can see how this can catastrophically contribute to aging processes. Um, and Our postdoctoral fellow, Tessafun Desale, uh, did an important research project on this where he asked the question, well, we know from previous research that depending on the cell type of origin that a senescent cell was before it went senescent, and depending on what process led it to become senescent, uh, cells can become more or less resistant to different senolytic drugs. So he asked the question, well, what about secondary senescent cells? Are they differentially susceptible or not to different senolytic drugs? And it turned out, to everybody's surprise, that not only were they resistant to some senolytic drugs, they were resistant to almost all senolytic drugs that we tested them against. This is a serious problem. So there's an entire category of senescent cells that sort of once you open up the barn door, you can't get back because you can't destroy them with the existing senolytic drugs. And of course, if you're 30 or 40 years old, you already have all kinds of senescent cells in your body, and this is a real problem. He developed an entirely different strategy by looking at the biology of these secondary senescent cells and comparing them to primary senescent cells, discovering that both types of senescent cells have aberrant iron metabolism. Some of this was already known in terms of that senescent cells accumulated very high levels of iron, and no one really understands why. Dr. Zasali really drilled down to that and discovered some of the processes that they were undergoing to do that, and by exploiting that the biology was able to develop two different categories of drugs that were effective both against primary and secondary senescent cells, broadly speaking. So this is a broad spectrum approach to destroying senescent cells uh, more effectively than any other kind of senolytic drug had previously been developed. Now, senescent cells did not evolve in order to kill us. As I was saying earlier, they evolved to do benign things like prevent us from developing cancer. And it turns out we now know that they also do a rather variety of other useful things for us, such as they prevent fibrosis, uh, and they play a role in regeneration following an injury. So it's a tricky thing that you don't want to be killing off all senescent cells all the time, but you obviously want to prevent them from accumulating. So trying to get that balance with small molecules is going to be kind of tricky. Potentially a better approach is to exploit the immune system. So the body already gets rid of senescent cells, but incompletely using the immune system. And one of our teams at SENS Research Foundation is working on how can we rejuvenate this ability that young animals have to clear out senescent cells or possibly fortify and use that ability through a cell therapy approach to remove senescent cells. The immune system has evolved for literally millions of years to do this and is possibly going to do this more smartly, more sensitively, pick the quote right close quote senescent cells to remove, and you leave the senescent cells that are still doing something useful if we can harness this biology. So this is potentially a better way to go about this, and they have been making good progress on that front. Um, in particular, we have known that a variety of different kinds of cells are involved in the process of senescent cell surveillance, as it is called. And in particular, uh, the starring role to date has been natural killer cells or NK cells. But it has not been clear that that is the most important in terms of what's going on in aging as opposed to in different kinds of models. Um, Dr. Sharma, who leads our senescence team at SENS Research Foundation, has just recently discovered a cell, which was predicted in this 2012 paper, um, that appears to be much more effective than NK cells at clearing out senescent cells, and that has the advantage from a biotechnology approach that it is much easier to harness for this purpose. I cannot disclose the identity of this cell or how it was discovered. We have to wait until it is published, but this is extremely exciting development and is going to really let us go after senescent cells in a more effective and I expect uh, a more sensitive way in terms of avoiding destroying senescent cells at an inappropriate time. So, senescent cells are just one kind of abnormal cell that accumulates in the aging body. Another kind of abnormal cell that accumulates in the aging body is cells with activated retrotransposons and transposons. So, these are the remnants of viruses that have gotten embedded into our DNA over deep ancestral time, and that with aging can become reactivated. So, we have an entire system of surveillance to keep retrotransposons suppressed in our cells. And that machinery becomes less and less effective as we age, and they start to reactivate. And this causes a variety of problems, including mutagenesis, when these transgenes, retrotransposons, reinsert themselves back into the genome, and also because they both drive senescence and are unleashed by senescence. So cells that have this damage into them are potentially really, really damaging to our bodies. Um, Dr. Andrei Gudkov at the Roswell Park Cancer Institute approached us some years ago with a proposal for a so-called retrolytic proof of concept. So retrolytic, going after retrotransposons through a destructive approach, similarly to a senolytic approach. So here we have a similar gene construct to what was originally used as a proof of concept for senescent cells, that as soon as a gene that is highly relevant to transposons gets activated, it triggers the destruction of the cell, just as the older proof of concept gene therapy destroyed senescent cells. And if this works, it would similarly prevent and reverse the aspects of aging that are caused by the activation of retrotransposons. Unfortunately, this has gotten frozen in time, no pun intended, um, because the transgene system that he developed that worked well in cells, he has not been able to get viable mice out of. And he made a couple of attempts at this and has not yet been able to get it to work. So he has asked for a no cost extension until he can figure this out. And as soon as he does, we will let you know. But the basic strategy here is very, very clever and something that we had funded and will fund again as soon as he is able to do it. There's a third kind of seemingly abnormal cell that accumulates in the aging body that we talked about in anti-aging that now seems not actually to be something worth going after. These are so-called anergic T cells. Um, the idea here was that certain kinds of T cells that recognize certain kinds of persistent viruses, most notably cytomegalovirus, CMV, um, because these viruses keep reactivating themselves, your body keeps producing more and more and more of the memory cells that are specific to tackling these viruses. And there was this hypothesis of limited so called immunological space, and that they were therefore metaphorically crowding out other kinds of immune cells out of the body. And that was contributing to the age-related decline in the immune system. Um, the evidence is really that that isn't the case anymore. That instead, really, there is a essentially infinitely expandable population of immune cells, and you do have large numbers of these so-called anergic cells, and that is a somewhat mixed blessing because they are still contributing to the control of these viruses, but other things are instead contributing to your lack of proper development of immune cells against other pathogens, only one of which is, of course, the fact that we're not producing them anymore from the thymus. Um, and here is some of the evidence that has been accumulated to show this idea of limited so-called immunological space is really not happening in aging. Um, So another category is glycosens. This is the cross-linking of functional proteins in our bodies and other kinds of damage to the extracellular matrix, the proteins that give structure to and support our bodies and allow them to function. Um, One of these is a cross-link. So when two adjacent strands of proteins get a sort of molecular handcuff that binds them together and keeps them from moving independently as they have to be flexible. Um, that contributes to a bunch of aging pathology, including possibly the stiffening of the large arteries and therefore damage to the heart as, and the kidneys and the brain caused by the increasingly intense pounding of the pulse on those organs. Um, in order to cut those crosslinks, you have to be able to produce them to do the molecular biology required. And SENS Research Foundation sponsored the research that first synthesized glucose pain to allow the reagents that we will need to deal with that problem. That was published in the journal Science some years back. They spun out a company called Revel, using SENS Research Foundation research and SENS Research Foundation funding, along with that of Kaizu, and they have developed prototype enzymes that actually cleave uh, glucosamine crosslinks. That will not probably work as an actual rejuvenation biotechnology because of the size of the molecules, but it's a proof of concept of the idea that you could cleave these suckers. Um, for reasons of time, I am going to skip over the next slide here, but that story is a bit more complicated than we would ideally have liked it to be, but still not insoluble. Um, Another kind of damage to the extracellular matrix is the fraying and calcification of elastin. So elastin is a protein that gives your arteries and other structures in your body, such as the lung, the flexibility they need to actually stretch just like an elastic band stretches. And as a result of a variety of processes, this protein actually becomes frayed, like a frayed shoelace, and no longer can do its function. And this fraying, both for chemical reasons and for biological uh, sort of regulated reasons, allows also elastin to become calcified, which further stiffens it and causes additional problems. So here I'm going to talk about medial elastocalcinosis. This is a kind of arterial calcification, but it is not the kind you are most likely to be thinking of. This is not the kind that you see on a coronary calcium scan. That is intimal calcification. That is calcification in the innermost layer of the blood vessel associated with atherosclerosis. Here we are talking about calcification of the elastin protein in the media, so one layer deeper into the artery. And this happens all across the vascular tree, not just in association with atherosclerosis, and it stiffens the artery over time and contributes to hypertension. It is a really serious problem. And at the time that we wrote the book, no one had even a strategy for dealing with this problem. Happily, scientists at Clemson University came up with a strategy based on this fact. Elastin does not appear as a naked protein, as it were, in the artery. It is instead surrounded by microfibrils of other proteins. And when it starts to fray, uh, those other proteins get torn away and elastin becomes exposed to the outside. That is what allows it to become calcified, but that also inspires this strategy. The idea here is you develop a nanoparticle that targets damaged elastin, uh, so that it can bind to damaged elastin that will only be exposed in the first place when that outer layer gets stripped away, when the uh, elastin has actually been damaged. And so using these nanoparticles, you can target drugs directly to damaged elastin and only to damaged elastin. And so this allows something that people have been talking about for many years, but not doing very effectively, which is bringing uh, drugs directly to elastin and repairing it. So here you see uh, a graph that shows, I can't see this as clearly as I would like. Um, these nanoparticles on the left here do not have anything associated with them, and the elastin remains damaged. Uh, in the uh, middle, sorry, second to the left, you see binding that to a drug that can. I've forgotten what this does. Hang on a moment. Oh gosh. Okay. Sorry. This is a drug that inhibits matrix metalloproteinases. These are some of the enzymes that are doing the damage to the elastin. So that has a very slight effect on the damage to the elastin. In the third uh, column there, you see targeting it with polyglycyl glycerate, which can stimulate the development of new elastin, and that starts to actually repair the elastin. And in the last column, you see a dual strategy where one set of nanoparticles actually remove some of that calcification by putting the chelator EDTA in the nanoparticle so that it directly removes the calcification locally, but again, only at the places where the elastin is damaged. And then you chase that with PGG and do the repair. And there you see an almost complete clearance of the calcium, a improvement of the structure of the elastin itself, and a return to normal structure. And I should be clear, that was in vivo in mice with a model of elastocalcification. So the next category we are going to go through is lysosens. This is the accumulation of damaged proteins and other materials inside of cells, unlike amylosens, which is talking about damaged proteins outside of cells. Um, so there's a couple of spin-out companies that were developed from research done in one case in SENS Research Foundation. So here they are, we're talking about atherosclerosis. So atherosclerosis develops because uh, LDL particles and other atherogenic lipoproteins become retained in the arterial wall. This attracts immune cells called macrophages in to protect the arterial wall from the direct damage that those trapped lipoproteins can do. The problem is that the modification of those lipoproteins inside the macrophage or before they take them up causes the macrophage to become poisoned and dysfunctional, and the macrophages themselves become trapped in the artery wall. And this is what leads to the development of the foam cell, and then the early stages, and then the more advanced stages of atherosclerotic plaque. Um, so Cyclarity, this spinout that was previously known as Underdog Pharmaceutical, they have an approach using these dimers of cyclodextrin. So these are carbohydrate-based molecules that are basically barrels that can scoop up different kinds of molecules inside of them. And by using this dimer-based approach, they can trap a kind of damaged cholesterol called 7-ketocholesterol inside of their structure and actually exfiltrate it out of the cell and allow them to return back to normal function. So what that potentially allows for is you remove the damaged cholesterol from the macrophages. The macrophages can return to normal function. They move out of the arterial wall, and then the normal repair processes allow the arterial wall to return to normal and actually reversing the atherosclerotic process. There is a different approach which we have also been involved in the initial founding of, uh, called Repair Biotechnology. This spinout, they are using a different approach. This is based on the observation that mycobacteria, so the species of bacteria that cause, amongst other things, tuberculosis, they live inside your cells by actually feasting on cholesterol inside macrophages, inside of lysosomes inside your cells. Um, and this is something that our own cells don't have the ability to do. And that is a good thing because you would not want to be sort of going around willy nilly destroying cholesterol. Cholesterol itself is an essential molecule that contributes to cell membranes and steroid hormones. But when damaged cholesterol is trapped inside your lysosome, that is doing you no good. And we don't have the evolved ability to uh, cleave and destroy and reuse those raw materials. But mycobacteria do, and that is what they do. They hang out inside your lysosomes and feast on that cholesterol inside your cells. Uh, Up in the upper left, you are seeing their colocalization inside of the phagocytes inside your cells, inside your body. Um, and so this company uh, broke down the enzymes that are responsible for doing that and were able to show that they could open up this cholestain ring that is the key uh, cyclic ring inside cholesterol molecules, sterile molecules that we haven't otherwise been able to crack as humans. And that they have shown in mouse models can reverse atherosclerosis in mice with established atherosclerosis in their arteries by about 48%. And if that can be done in humans, we are on the verge of a revolution. We are already, you know, damage repair aside, SENS aside, we are already in the midst of a revolution in atherosclerosis, where between PCSK9 inhibitors and the upcoming development of obicetrapib and other drugs, we are potentially looking at being able to prevent people who are 30 years old or so from ever developing atherosclerosis, provided they are non-smokers. And if we can combine that with actual damage repair, if either the repair biotechnology approach or the cyclarity approach actually goes in and reverses this underlying pathology inside of macrophages, uh, we could eliminate atherosclerosis entirely within a shockingly short period of time. A different kind of intercellular aggregate is called lipofuscin. This is actually one of the earliest forms of intercellular aggregate that was discovered back in the 1950s. Um, and a major problem that we have had since that time up until just a couple of years ago is that everyone trying to study lipofuscin was not really studying lipofuscin. They were cooking up artificial lipofuscin because lipofuscin itself was so difficult to isolate out of cells. Every method used to extract it would damage it in ways that made you not really clear that you were realistically replicating what lipofuscin would do inside of a cell. And also, it was just so difficult to get out that you could never get enough quantity of it. Um, our partner, Dr. Grune, in Germany, has developed a new method for extracting lipofuscin from cells and getting it in quantity, and has access to both human tissue and to large numbers of heart tissue from horses, and both therefore has enough raw material and a method for extracting it that it can actually work now directly on lipofuscin. And SENS Research Foundation is funding him now to use the classic uh, lysosens approach that Dr. de Grey originally developed back in the early 2000s, which is using the power of bacteria from soil to identify enzymes that can do what our own cells cannot, which is break down the essential bonds inside of lipofuscin and then allow the cell to do the rest of the work thereafter. He has already made an important amount of progress on this. Using a mixed soil bacteria sample, he can now uh, break down lipofuscin very substantially and release fluorescent breakdown products from that. What he next has to do is identify the specific enzymes that are responsible for this and figure out which ones do the most important steps in that process so we can turn that into a rejuvenation biotechnology that we can put inside a person's cell to do it in situ. An important property of these enzymes that he has discovered is that these enzymes are not active inside the bacteria itself. They are actually released into the bacteria's environment and break down the lipofuscin outside of the cells, and then they take in the broken down raw material and digest them further. And that has pros and cons, but the pro is that those enzymes are easily accessible and don't rely on the specific intracellular conditions of the bacteria to be active. So that is very, very promising stuff. Um, Another project here is targeting intracellular tau. Now, tau is a perfectly normal protein that is involved in the shuttling of materials inside the cell, but when it forms aggregates inside the cell, it contributes to a variety of neurodegenerative diseases related to aging. Um, and the problem is there are a number of people who are now, I mean, I should say it's a problem, but it is not the worst kind of problem. They are going after tau aggregates using a damage repair approach. That is good. Many companies now have antibodies designed to remove abnormal tau from the brain. The problem is those antibodies can only remove it from outside the neuron, and it is inside the neuron where aberrant tau does most of its damage. Outside of the cell, what you will mostly be doing is preventing that tau from being transmitted into new neurons, but you will not be rescuing the neurons that are already affected by aberrant tau. Dr. de Grey spotted this paper that is up in the upper left-hand corner of the slide there, where these scientists used single-stranded oligonucleotides, not oligonucleotides, single-stranded, my mind has gone blank. I apologize. Single-stranded uh, nucleic acid material, anyway, um, that had phosphorothioates. So here on the left, you see a normal nucleic acid sequence is composed of phosphodiester bonds. Uh, Here, they have a modification that bacteria sometimes use called phosphorothioate, where instead of a phosphorus attached to an oxygen, you have a phosphorus attached to a sulfur. And by using a single-stranded version of this, you can actually target antibodies inside of cells. So Dr. Gray thought, um, well, we can do even better than that. We can target, instead of regular antibodies, again, antibodies that just bind to something and pull it away, we can target these catabodies, these catalytic antibodies that can go in, break apart into small pieces their target, and then come back to the well and break another and another and another and another and clear it out entirely. This is all conceptual at this point. We haven't done any of this. Our scientists have been working on this. This is Dr. Sharma and Dr. Manikan. Um, but we are making uh, good progress along doing this, and hopefully we'll have a working model in cells before too long. Um, Mitosens. So the cell accumulates, the body accumulates with age a small number of cells that have these large deletion mutations in them. And once these large deletion mutations occur inside of a cell, those mutations, mitochondria bearing them, rapidly overtake the cell entirely, so that there is nothing left but mutant DNA cells inside. Um, and how are we going to deal with this? Well, uh, the strategy is what is called allotopic expression. The idea here is it is hopeless to try to repair each and every one of those mitochondria by doing gene therapy on each of those many mitochondria inside each cell in the body. Uh, Instead, we are going to preemptively place copies of those genes inside the nucleus of the cell where they are prevented from the damage that they are caused by the operation of the mitochondria themselves. And thereby, we will be able to express the proteins that the mitochondria needs from the nucleus uh, produce the proteins themselves in the main body of the cell, the cytosol, and import them into the mitochondria. And then the mitochondria will be able to operate properly because it will have all the components that it needs to generate energy, even if the mitochondrial genes themselves have become mutated. SENS Research Foundation sponsored a strategy back in the mid 2000s using this so-called co-translational import strategy, whereby we were going to make it easier for the mitochondria to take in the proteins that are produced in the cytosol by targeting them very, very close to the surface of the cell. And at least in the hands of this group in Paris that we sponsored to do this research, they were able to get this mitochondrial protein called ND4 into the mitochondria, which had not been effectively done before. And they were able to go one step further than this by doing it in vivo. So they targeted both the wild type version of the gene and a mutant version of the gene into the eyes of mice, and were able to show that the mutant version of the protein caused visual impairment, and then putting in the normal form of the protein rescued that visual impairment. You can see in the middle here this test that they use to test that. It's a rotating box, and you can imagine this sort of kaleidoscope moving around and around. And the test is watch whether the mouse follows the rotating circles or not in each eye. And sure enough, when they put the mutated version of the protein into their eyes, their tracking got worse. And when they put the wild type version into the eye afterwards, it got better again. And this was actually taken one step further into human trials for people who have uh, a mitochondrial. Oh, goodness, I haven't been advancing my slides properly. Hang on. Oh, someone is taking care of it. Thank you. um,
I'm able to move the
good.
slides, but you are on the
Yeah, yeah,
GenSite trial?
yeah, good. So
Doing
yes,
awesome.
we have.
I'm loving it. I
Sorry.
just want to say I'm loving it. Sorry. Go ahead.
Thank you. I am stumbling all over the place, so I'm glad you are still loving it. Um, GenSite, this company, took this into human clinical trials. These are patients who have a mitochondrial disease called Leber's hereditary optic neuropathy. Um, this causes them to eventually lose their sight and become blind. They turned this into a rejuvenation technology, inserted the gene into their eyes, and caused them to have a significant amount of recovery of sight. This is the number of uh, letters you can see on an eye chart. They progressively improved, and this is after they had stopped delivering new therapies. They give these people the therapy in one case after they had lost sight for 6 months, in the other case after they had been losing sight for a year, and then follow them up for up to a total of 5 years. And throughout that entire time period, their vision got better and better and better. I have bad news for everybody, which is that they have been unable to actually get a licensed therapy out of this because they have not been able to scale up what was originally a tiny, tiny pilot scale because they were dealing with very small numbers of patients. They have been unable to scale that up into a good manufacturing process quality product that they could deliver to people as a therapy. Um, and therefore have been stalled and unable to turn that into a human therapy. I really hope they are able to overcome that problem because, at least in their clinical trials, they were doing miracles for these small numbers of people. And it is through exactly this kind of process of targeting these things for people who have inherited mitochondrial disease that we will eventually get therapies that we can use for people who have mitochondrial disease because of aging instead of mitochondrial disease because of inheritance. Meanwhile, our team at SENS Research Foundation has been making excellent progress on the same front. up until our team stepped in, there were still a lot of reasons to doubt even the ND4 result that I was showing you a moment ago, the mouse work, because the rescue in the cells of the activity of the electron transport chain had never been totally convincing. Our team at SENS Research Foundation, led by Dr. Dubumanatham, um, was able to successfully do this with the genes ATP6 and ATP8 in cells, convincingly show that yes, indeed, uh, these cells, first off, have the mutation in the first place, and secondly, restore normal function after we deliver our transgene into these cells, that it really is being produced as a protein in the cytosol, that it really is being introduced into the mitochondria, incorporated into the electron transport chain, and rescuing function. Um, and they have subsequently made progress to one degree or another against every single one of the other 11 mitochondrial encoded genes. And we are now doing this in living mice. So this is using a technology that Dr. Gray was farsighted enough to develop in advance, knowing we would need it eventually, which is mice that are much more easily manipulated to have a transgene of arbitrary size into their cells, and introducible potentially at any point in life. Now, in this case, we have done this more or less in advance rather than um, late in life as we eventually plan to do. But the key here is that we have developed mice that have an inherited mitochondrial mutation, and that we are able to rescue their mitochondrial function by activating an allotopically expressed gene that we have engineered into their nucleuses. And that, I hope, will be published in the scientific literature very shortly. These mice, unfortunately, do not have a very dramatic phenotype. And so what you will see when this is published is not, a decrepit mouse that suddenly is up dancing the Macarena, but we will show at the biochemical level that these mice's cells are operating much more healthily than they were without this transgene to rescue the function of their mitochondria. Oncosense. This is making the body invulnerable to cancer. Um, Here we have sponsored a couple of projects. The core strategy that Dr. de Grey came up with shortly after the development of the original SENS paper in the Annals of the New York Academy of Sciences is what is called WILT, whole body interdiction of the lengthening of telomeres. And the idea here is that although telomerase does not cause cancer, that is not the claim, the lengthening of telomeres by one mechanism or another is an essential part of cancer actually becoming life-threatening. And so you have a problem, which is if you do not stop that process from happening, the body will eventually develop cancer because of mutations, and you will die of cancer. If you stop that from happening entirely, the body will eventually die because it can't renew its tissues anymore. But what if we can combine those things? What if we take away the ability of cells to lengthen their telomeres preemptively so that cancer can never get started in the first place, but we periodically replace the cells in the body by renewing all the tissues with engineered cells that have had their telomere maintenance machinery removed, but have nice long telomeres that we have lengthened in the lab in advance. So you have a long sort of a shelf life to those cells in the body. And you can thereby keep ahead of the denuding of your cells and tissues of the cells that they need while keeping cancer indefinitely at bay. As a pilot to show the basic idea that such cells could rescue that kind of tissue atrophy, SENS Research Foundation sponsored this research in the lab of Carl Leonard Rudolph, one of his, at that time, graduate students, Zhenyu Zhu, showed that if you had mice that had very, very, very short telomeres and were otherwise going to live very short lives because of tissue atrophy, if you then introduced cells from other mice that had longer telomeres but had no telomere lengthening machinery in them, that you could partially normalize their lifespan. This was not a perfect uh, proof of concept for the idea, but it was enough to show that the basic idea was feasible. We also had a project at SENS Research Foundation for several years working on a better assay for the ALT mechanism. This is an alternative lengthening of telomeres mechanism, ALT, which is a way that cancers sometimes lengthen their telomeres without um, using the telomerase mechanism. And the idea here was if we could develop a much simpler way, a much faster way, a much cheaper way to measure alt activity, we could spark a lot more research into the alt mechanism, which at that time was not being properly followed. Um, I have to say, we didn't really succeed in this. Another lab was able to get a equally good assay as we could do. We eventually partnered with them, and both labs were able to contribute to that progress happening, but it was not enough to spark the revolution in alt research that we would have liked to do. Um, so we still need to make a lot of progress in that area, but some progress has been made and we have contributed to it. Um, so, surprise, cancer has not been conquered in the last 10 or 15 years, but I will say just in the last few years, we have started to make, let me take a step back. Um, this is a process that Dr. de Grey has sometimes characterized as Wilt2. This is a molecule called 6-thio-2-deoxyguanosine, or thio, which is being developed by this company, Maya Biotechnology. Um, This works a little bit like the antiviral drug valsiclovir, if you're familiar with that. So um, this is an analog to the nucleoside guanosine, which gets preferentially misincorporated into telomeres by the telomerase enzyme. Um, so uh, telomeres uh, notoriously have a TTAGGG structure that just repeats over and over and over and over again. Um, and so it has a lot of guanosine in it. And so the fact that the that the telomerase enzyme, quote unquote, prefers to incorporate thio instead of actual guanosine is an exploitable point because once it has incorporated guanosine, the telomere will become dysfunctional. You will get uh, the so-called TIFs, telomere damage induced foci in them, and the telomere will trigger damage to the cell and eventually the destruction of the cell, and thereby prevent and the cancer from developing and stop the process in its tracks. Um, Dr. de Grey is very enthusiastic for this. I will confess I am not very enthusiastic for this. I think the cell is likely to be able to mutate its way around this the same way it mutates around every other kind of molecular based strategy against cancer. And in fact, there is already evidence for this. Here on the right, you can see that out of a group of 77 non-small cell lung cancer lines that were tested, there were 4 that were invulnerable to thio. And the reason turned out to be that these 4 lines were expressing much lower levels of a solute transporter called SLC3 43A3, which is a nucleobase transporter. So by not incorporating nucleobases rapidly into the cell, it was largely able to exclude thio and rely on its own resources, and therefore was not affected by it. And it is not difficult at all to imagine that when you give this to a person that has a tumor with millions of cells in it, that there is going to be at least one such cell that has some version of this mutation in it that is going to allow it to exclude thio from inside of it, and the other cells will die off and it will prosper and multiply rapidly and become the dominant clone, which is exactly why every other kind of chemotherapy fails. And this is just one example that Jerry Shea has discovered. I can assure you that clever as Jerry Shea is, he nor I nor anyone else is nearly as clever as evolution is, and what cancer is an evolutionary disease. It will figure out a way, I expect, around this therapy. So we really need to go back to the original Wilt strategy and get this job done. And we have not yet done that. But I will say, in the last few years, we have gotten some damn fine consolation prizes. So this is something some of you may have taken notice of. The company BioNTech, the same people that gave us what is usually referred to as the Pfizer COVID vaccine, use the same mRNA vaccine technology to develop personalized cancer vaccines for people with pancreatic cancer. And when I say pancreatic cancer, um, there are kinds of pancreatic cancers called pancreatic neuroendocrine tumors, which are not very dangerous at all. Here we are talking about pancreatic ductal adenocarcinoma, PNAC. These are deadly. 88% of patients who develop any stage of PDAC will die of it. BioNTech was able to take tumor samples from patients with PDAC cancers, develop personalized mRNA vaccines against them, immunize these people with them, and so far, through all the follow-up that they have done on these people, uh, the people who developed a immunological response, in other words, they were able to show that the T cells of these people were activated in response to tumor tissue. None of them have shown any progression of their cancer at all uh, across 18 months of follow-up since they received the vaccine, which is just completely unheard of. Now, this is a small number of patients. This is not, 10-year survival. So I don't want to say we have shown that we can cure PDAC, but this is seemingly more progress that has been made in PDAC than in the history of medicine. So this is incredibly exciting against a really hard cancer. Um, Meanwhile, some of you have seen this other remarkable advance in mismatch repair defective rectal cancer. These are patients that had rectal cancer. They have a defect in a form of DNA repair. Lots of these people were actually born with this DNA defect. That was what made them vulnerable to cancer in the first place. And out of the 12 patients that they treated with this, uh, every single one of them is still alive. They have shown an overall response to this drug in every single case. This is a PD-1 inhibitor antibodies. So these are so-called checkpoint inhibitors. These are drugs that prevent the cancer cell from using a so-called cloaking device, whereby they normally say nothing to see here when the immune system comes looking for them. These drugs come in and turn off that cloaking device and allow the immune system to so-called see uh, the immune cell. And so far, that has allowed them to wipe out their rectal cancers with essentially no serious side effects in any of these patients. Again, this is a small number of patients. This is not 10-year survival. We don't know how far this is going to go, but this is incredible progress. The other caveat I should add is that this is locally advanced cancer. This is not metastatic disease. which is where we have had the hardest problem making progress against cancer. So, you know, we still need to make progress, and I still predict we are going to need wilt or something like it to conquer cancer. But this is remarkable progress we have made. Um, I didn't even develop a slide for it, but folks should look into tumor treating fields as another quite remarkable advance that has been made just in the last 5 years. Replenish-SENS. So this is the replenishment and replacement of cells and tissues. Um, Obviously, we lose cells from a variety of tissues as we age, and the only thing you can do about that is to replace them. Um, an incredible advance that was made after we published Ending Aging is, of course, cellular reprogramming. Anyone interested enough in the entire subject of medical intervention in aging will know about this by now. This is an absolute cause célèbre. It got the Nobel Prize in Physiology or Medicine in 2012. This is still kind of astonishing biotechnology whereby scientists can take a cell that is fully differentiated, such as a skin cell or a B cell from your immune system, and wind the developmental clock back on them to a pluripotent cell, in other words, a cell that has the developmental potential to become any kind of cell. And then by using different chemical and biological cues, turn that cell into a different kind of differentiated cell uh, and use that cell for therapy. Um, Incredibly exciting technology. Um, it's being used in a variety of contexts. So one project we have at SENS Research Foundation is we are funding Dr. Jean Hebert at the Albert Einstein College of Medicine with this amazingly promising strategy he has for repopulating the neocortex. So the neocortex is the part of the brain that most makes us human. It is where we keep our uh, memories, our identity in the brain. We absolutely want to hold on to the neocortex. Of course, we want to hold on to the entire brain and the rest of our bodies too. But this is clearly just critical to what it means to be human. No one has had a realistic strategy for doing that up until Dr. Hebert came up with his strategy. So um, an important thing to understand is that if you think about the way the brain looks when you look at it, it's all folded up. It has those invaginations in it. um, the neocortex alone, if you were to spread it out over a table, it is 2,600 square centimeters. That is roughly a square 23 inches on a side and 3 to 4 millimeters thick. um, it was only in the last 5 years or so that scientists were able to inject locally at a tiny area in the brain um, a small number of neural precursor cells and show they could survive and integrate locally. If you look up in that figure up in the upper half of the screen, you can see that just a fraction of a millimeter away from the injection site, you no longer have any new neurons inside the brain, right? So that you only get these neurons locally where you inject them. I mean, you consider the size and the folding of the neocortex, that just won't scale. You cannot keep injecting over the course of an extended lifespan, cell after cell after cell after cell over this huge space and depth of the brain without doing horrendous damage to the brain. So simply injecting neural precursor cells into the brain was not going to work at scale. Dr. Hebert's strategy was quite different. He said there is a different kind of cell in the brain, the microglia, which we already know from other research. If you destroy the microglia, other microglia will expand and repopulate across the brain. Um, they are constantly so-called tiling. They are out on patrol looking for places where there are not microglia and replacing them by getting into that space. So what if, said Dr. Hebert, we were to inject the brain with a drug that would destroy a significant portion of the microglia, and we were to replace those microglia with engineered microglia, that we could then give a drug that would activate a cassette inside the microglia that will allow them to be reprogrammed into neuronal cells instead. Well, then you have a way of spreading the engineered cells all across the neocortex and then turning them into neuronal precursors and scaling all across the brain the repopulation, the integration into local neuronal circuits of new neurons. And that would be a strategy for maintaining the neocortex potentially indefinitely. This was incredible out of the skies strategy to do with this. And he has made quite significant progress with SENS research funding over the course of the last several years. So he has already, this is a actual picture of engineered microglia spread across the brain. And he has now tested a range of concentrations of different drugs, the first of which targeted a survival factor inside microglia called microglial colony stimulating factor 1. These drugs can destroy the native microglia, but the engineered microglia survive precisely because they've been engineered to survive these drugs. So he has been experimenting now with a variety of different delivery methods and different drugs that would allow him to destroy enough of the old microglia to make room for the repopulation while still leaving the engineered cells harmless. They have also gotten larger harvests of donor microglia. Again, this is so far all in mice, and developed a better so-called shielding system to protect the transplanted neurons from the drug. He has also developed a screening protocol which allows him to select microglia that have successfully integrated the shielding system. So obviously, you don't want to put, 1 million cells into the brain only to discover that 300,000 of those cells do not actually have the shielding system and immediately die upon encountering the drug. So he's got a better system for selecting out only those cells that have selectively incorporated the shielding system. He has also improved his reprogramming, at least in vitro, protocol for the microglia, so that he is now approaching 5%, which is a very high level of reprogramming efficiency. So making very good progress along this way. And if we can get all those pieces to work together, we could keep the neocortex running, potentially indefinitely, at least as far as that goes. And when you combine that with removing tau, removing beta amyloid, the brain could really be maintained in a youthful state for a greatly expanded period of time. Meanwhile, this biotechnology company, Blue Rock, has developed a way of harvesting embryonic stem cells and turning them into dopamine producing cells for transplantation into people with Parkinson's disease. So they have successfully differentiated these cells into dopaminergic neurons. They have just in the last 2 weeks completed a phase 1 clinical trial where they implanted these cells into the brains of 12 subjects with advanced Parkinson's disease. We don't have any data aside from a brief press release from the company, so I can't give you a lot of details on this, but they say they have confirmed the feasibility of transplantation and they have got evidence that the cells have survived and have engrafted into the brains of these patients. And they are expecting to have a phase 2 clinical trial started in the first half of next year. And they will announce the results of this phase 1 clinical trial more fully at the International Congress of Parkinson's Disease and Movement Disorders, which is in Copenhagen at the end of this month. And this happened in the depths of the pandemic, and so a lot of people unfortunately missed it. And it is really a tragedy because this was an amazing, amazing development. So here, these scientists at the Dana-Farber Cancer Institute and Harvard and affiliated centers worked together. The person on the left is a guy named George Lopez, goes by Doc. He's a biomedical entrepreneur that made his original fortune developing biomedical devices. He unfortunately developed Parkinson's disease and got tired of having Parkinson's disease and donated, having heard about reprogramming cells and the early successes back in the 1980s and 1990s with clinical trials using field-derived cells, mixed populations in Parkinson's disease patients. He wanted this to happen in time for him and donated $2 million to scientists at Harvard and Brigham and Williams to get this happening for him personally. So he had uh, these scientists take his own skin cells, uh, turn them back into induced pluripotent cells, uh, transdifferentiate further forward into dopamine producing cells of the sort found in the substantia nigra, which is the area affected by Parkinson's disease, was able to show that these cells were not immunogenic in mice and that they would uh, engraft in mice successfully into the substantia nigra. He then, there's a story in Stat News about this. It's quite remarkable, sort of the process where they had to take these cells at the Dana-Farber Cancer Institute, derive them from doc, and then put them in coolers and rush them via medical helicopter to Brigham and Williams to actually do the transplant into doc. Um, they got him into his brain. He did not require any immunosuppression because they were his cells. This is the miracle of induced pluripotent stem cells. Using PET, they were able to show that they had new implanted dopamine producing cells in the relevant area of his brain, in the putamen. This is what you see here on the right, that those cells were still alive 18 to 24 months after implantation and still producing dopamine, and that he has had some improved clinical function. Well, excuse me, I shouldn't overstate the case. His clinical status has been stable over the 18 to 24 months since his implantation. Um, this was published in the New England Journal of Medicine in 2020, and therefore everyone was a little bit distracted to hear about it. Um, COVID had another negative effect, which is that it stopped them from undergoing a clinical trial, which they had planned to undergo when this all happened. But I am assured by personal contact with the surgeon that implanted these cells into doc and will be the lead surgeon for the clinical trials, that the clinical trial is indeed going ahead. So this is really happening. We are soon going to have the cell side of Parkinson's disease potentially uh, under control. Now, we've been talking about the SENS 7, and there is a question always of, is there an eighth category of SENS damage? Potentially there is. This is something that Dr. de Grey and I both actually agree on. Um, this is persistent viruses that accumulate in our cells with aging. I've already mentioned cytomegalovirus. Herpes simplex virus is another example. The zoster virus that is responsible for shingles late in life, or at least late in current lifespans. All of these things are caused by persistent viruses that the body does not successfully clear. The reason why Dr. Gray has never formally declared this an eighth kind of aging damage is because classically, when we talk about aging, we are talking about things that the body inflicts on itself, right? Like obviously, if you have a shard of glass, that is implanted into your flesh and cannot be safely surgically removed, that is bad, but that is not aging, right? And so viruses, in a certain way, are analogous in as much as they are completely alien to the body. But let's not be silly. If we are going to conquer aging, we want to remove that shard of glass. We want to remove those viruses. Um, and so that is what we're talking about here, the clearance of viruses out of the body. Um, and this is potentially responsible for more than just the obvious infectious causes that we know about. For instance, a lot of you will have heard about the so-called viral hypothesis of Alzheimer's disease, for which we just had a powerful additional line of evidence in the last couple of months in preprint form that the shingles vaccine actually seems to dramatically reduce the incidence of Alzheimer's disease in a natural experiment. This is not a classic epidemiological study. This was a random allocation of people by sheer accident in Wales to shingles disease, to shingles vaccine or no shingles vaccine. Seems to have demonstrated that, at least in women, the shingles vaccine has prevented very large numbers of people from developing Alzheimer's disease. And so the best solution to that is, to remove the virus. And so the elimination of pathological viruses in aging is likely something that we are going to want to deal with as part of the SENS program, and therefore is definitely an important thing that we didn't cover in ending aging, and that we would certainly cover in a follow-up if we were writing one today. And of course, there's been the progress of SENS Research Foundation itself. This includes the dramatic improvement of our laboratory facilities. If you look on the left there, that in the yellow, that was not even our first lab. Our very first lab was a garage owned by John Schloendorn in Arizona. Then we had a tiny little, not very impressive facility in Sunnyvale. Then we had this relatively small chunk of this building in Mountain View, California, which is represented in yellow on the floor plan there. Within about 5 years, we doubled that out, first expanding to that pink area, then to the blue area. And in 2022, of course, Gennady was there for this event. We had the grand opening for the further doubling of our facilities, which, amongst other things, allowed us to introduce our vivarium, which is what you see in the big picture on the right. So previously, any animal work that we did, we had to do with partners in outside facilities. Now we actually have lab animals who are experiencing rejuvenation biotechnologies in our labs right now. So that is really important progress. We also have fancy new kit that we did not have, that in some cases didn't even exist when we first established SENS Research Foundation back in 29, let alone when we wrote the book in 2006. We are not there. Aging is not conquered, but we are making progress, and we're getting there. So, yeah, I wish I had a more dramatic ending to share with you, but it's dramatic progress nonetheless, and we are going to get there. We just have to keep working at it. Thank you.
Yes, thank you very much, Michael. This is indeed quite an impressive overview of the progress that has been made in the longevity field over the past 15 years. And now I would like to open this up for questions. We have about 16 minutes for questions. So let us start with Dr. Jose Cordero.
Hello, Michael. This is a fantastic presentation indeed. And let me tell you that your book was very influential in me, and I helped with the Spanish translation. And reading your book, then I decided to write another book. And I have my fantastic co-author here with me, David Wood. And so we published our book in Spanish 5 years ago. And in English, it just came out. And many things have changed in 5 years. So imagine what has changed for you in 15 years. But 2 quick questions. How did you split the work between you and Aubrey? That's question number 1. Question number 2 is that Ray Kurzweil has been making projections for half a century. And by the end of last millennia, he gave 2 magic dates, 2 really magic dates that are in his new coming up book that will come out next year. The singularity is nearer. The dates he has kept for almost 3 decades without change. He said, in 2029, we will reach longevity escape velocity. That means if we make it to 2030, we will basically live long enough to live forever. And he's 75 years old. And the second magic date is 2045, when we will have rejuvenation technologies. So those are my two questions. How did you split the work? And how about projections for longevity, escape velocity, and rejuvenation?
So I would say that at SENS Research Foundation, we are working principally on the development of new rejuvenation biotechnologies and the improving of rejuvenation biotechnologies. The principal work that Dr. de Grey is working on at the Longevity Escape Velocity Foundation is on combinations of existing both rejuvenation biotechnologies and other aging interventions, some that are the classical geroscience type approaches to achieve this goal of robust mouse rejuvenation. In other words, taking a mouse that is already in late middle age and doubling its remaining lifespan. So he has a very sophisticated way of doing combinations of different longevity interventions in mice in a way that will really answer the question, first off, what are the combination effects going to be, and whether the effect of any particular intervention in that cocktail is good or bad. So if there is a deleterious effect, if you do a combination only once, you will never know which part of that combination did the harm. If, on the other hand, you greatly extend the lifespan of the animals, you don't know which of those several interventions did the work and which are just passengers. So he has this very sophisticated method of doing essentially double controls throughout that process, so that at the end of the experiment, we will really know who did the work, positive and negative, and therefore which should graduate on to the next step to yet further improve the progress that he has made. So it is very sophisticated work. I would expect nothing less from Dr. de Grey. He is an amazing scientific mind. In terms of the dates, I have always been reluctant to put dates on things. I think that science happens at the rate science happens. You can make science go faster than it would by investing more money, more people, more resources into the problem, but you cannot predict on a timeline when breakthroughs will happen, let alone when complex breakthroughs like longevity escape velocity will happen. I will say I am pretty confident it is not going to be by 2029. But beyond that, I would not prefer to predict on it. I will only say that they will happen faster the more resources are poured into the program. So everybody out there, donate to rejuvenation biotechnology organizations, invest in rejuvenation biotechnology startups, and please lobby your politicians and tell them you want more dollars put into biomedical research, and in particular into biomedical research against the biology of aging, because that is how we are going to conquer this thing.
Yes, indeed. Thank you very much, Michael. And this kind of message is definitely one that the public needs to hear, that it is up to us. It is up to how much we are willing to commit and invest in this field in terms of how much progress we achieve. So next, I would like to go to Dr. Bill Andrews for any questions or comments that he might have. I'm particularly curious, and several of our audience members are curious to hear his thoughts about the wilt approach to fighting cancer.
Well, my questions won't have anything to do with the wilt, and these presentations.
I don't have to run. Good.
Okay. No. It's there's a lot of plus thoughts about wilt. I don't want to condemn the whole thing. I think there's some problems too that have to be resolved, but I believe Aubrey can get those resolved. But my questions are really related to the senolytics, and I've got kind of a two-part questions, both related to senolytics. Let me just, the first question is, I'm going to go to an extreme here just to kind of make the question clear. Um, when a person's about 100 years old and they've got really skin that's really thin and fragile, is it known what percent of the skin stem cells are already senescent?
So unfortunately, there is no single biomarker for senescent cells, as you probably know, and this makes it very difficult to get a hard number on how many cells are senescent, even in a mouse, let alone in a human. So when we say that there are a number of senescent cells inside of a mouse or a tissue or a cell population, what is typically being done, at least if the scientists are being rigorous, is they are sort of layering on a Swiss cheese of, let us test for the level of protein of P16. Let us test for the level of a DNA damage marker called gamma H2AX. Let us test for the production of SASP factors, these inflammatory factors and other enzymes that are released by senescent cells. And by stacking those all together, let us try to get an estimate of the number of senescent cells inside of a tissue. And that just quantitatively makes it very, very difficult to get a realistic impression in any tissue, even in a mouse, of how many senescent cells there really are in the tissue, let alone in humans. And, for reasons you will understand, there has been just a lot less testing in this regard in human tissues than in mouse tissues because people are less enthusiastic than scientists are about taking widespread samples from aging humans, especially centenarian humans, who, by definition, they're old, so they're more fragile, so they have less, regenerative capacity, to be taking out, large numbers of biopsies to get a representative sample from a representatively large number of people to get an idea of the range. So the simple answer is we don't really have good numbers on any of that. We just have the general trend that it's more and more and more by any lab's assessment. And if you look at, there's been several populations, several groups that have tested this in their populations. They tend to be small sample sizes, relatively narrow age ranges, like say age 20 to 70 or so, and generally using not super robust ways of assessing it just because of limited sample size and so forth and so on. So
Okay, well,
yeah,
it
we
sounds like
don't have a,
sounds like the answer is you don't know. So let me
yeah.
rephrase the first question. Um, can you say that in a 100-year-old, the percentage of senescent cells is less than 50%? Or is that an I
I
don't know?
don't know. Yeah, I'm afraid that's an I don't know either. I mean, I've seen estimates, they're in the ballpark of 50% for some tissues in mice. So I wouldn't want to say it's crazy to think that half the cells in a centenarian skin might be senescent. Um, it sounds like intuitively, it sounds like, well, that can't be right. Um, but
Might be 50%.
I am saying that out of intuition. I am not saying that out of data.
So my concern is that senolytics could be lethal to a very old person. I mean, it'd be like burning a person's skin off. A person can't survive without skin, and killing off, like, let's say 50% or more of the cells because they're senolytic might be a lethal event, especially when you don't have cells to replace them because.
Well, and you've just answered the question, right? So 100%. Like the proposal of SENS is never let us do one of these 7 things to one kind of aging lesion and leave it at that. And let's, by the way, wait until you're 100 years old to do it, right? The idea, again, if is there a way I could get my slides back up? Let me
Well, actually,
present
you
again.
already started on my second
Yeah.
question, but
Okay,
but go
so
ahead.
the idea here is we never want to be attacking only one form of aging, and we never want to be attacking it when a person already has, very, very advanced pathology. We are not, and Aubrey de Grey has never claimed that we are going to be able to take a 100-year-old and rejuvenate them and give them indefinite lifespans. The claim rather is that if we intervene early enough and repair damage comprehensively enough, we can forestall aging indefinitely. And so let me get back.
Okay, well, let me just say that I said my first question was going to an extreme. That's why I picked a 100-year-old, just to make my question
Right.
clear. The second question now deals with a 60-year-old. Okay, and so if you have, and so if we don't know what a 100-year-old is, we also don't know what a 60-year-old is. And a 60-year-old could have a significant number of senescent cells too. But ignoring that for a second, when you kill a senescent cell in a skin, let's say I'm using skin just as an example, but of course there are senescent cells everywhere. When you kill a senescent cell, isn't another cell going to have to divide to replace that killed cell?
Well, it's not simply a matter of taking up space, but you are right to raise the issue. So one theory that was actually advanced by a guy named Magnus Lynch back in 2009 was that part of the reason why senescent cells aren't all removed was exactly that because it was in a dangerous neighborhood, as it were, because of course, in most cases, if you have one damaged cell that is in danger of becoming danger of becoming cancerous, you will have other cells in the same neighborhood that are in danger of becoming cancerous or maybe even be cancerous, that you don't want to give them space to replicate themselves. And so maybe why senescent cells aren't cleared out is exactly in order to prevent cancer from spreading. And so you could theoretically say that you would expect more cells to replicate and expand. But the sort of error catastrophe that I think you were imagining happening, we have looked at that in mice. I mean, not we, SENS Research Foundation, but other scientists have looked at, and that does not seem to happen. You do see a clear reduction in the total number of senescent cells, and in some cases, you do see an expansion of cells that don't become senescent if you do it in a young enough mouse. But again, certainly we will need to be replacing cells as we remove cells if we want to get the full benefits of senolytic therapies. This is also why it's important to look at the immune process of removing senescent cells, both because that gives us proof of concept that this catastrophe doesn't happen because you're removing senescent cells throughout your entire life without this catastrophe happening, but also because it can be more sensitive about when senescent cells are removed and under what conditions and so forth.
Yes,
Well,
thank you, Michael.
Let me just quick, because the catastrophe is not what I'm referring to. The catastrophe I'm referring to is that when another cell divides to replace that cell, you get closer to the Hayflick limit. And as a result, you might be accelerating aging by using senolytics. And what is your comment on that?
Yes. So this is
and the answers could be shorter, Gennady. The answers could be shorter if we were running out of time. Good.
Yeah. So I'd like to fit in a brief question from David Wood as well, because we only have about a minute and a half remaining. So Michael, a quick answer, maybe 15 seconds, and then let's go to David.
That question was actually what I was trying to get at with this catastrophe problem, which is the idea of cells in random area dividing and becoming senescent, right? That's the whole point about the Hayflick limit, is the cells become senescent. So the fact that you get a net reduction in senescent cells and don't see a progressive increase thereafter is pretty good evidence that this kind of catastrophe of increasing senescent cells by decreasing senescent cells doesn't seem to happen. But again, the real solution to that is to replace those cells with new cells that will be able to take up that space and do the job they were doing and prevent this replication problem.
That's the solution. If you guys can figure out how to do that, yes.
Okay, let's go to David.
Fascinating information, Michael. Very encouraging. As well as keeping track of what's happening in all these different areas of SENS, are you also keeping track of where you think the biggest problem lies, the area where most attention is needed, where you are kept awake at night because you're not sure that the solution is at hand?
Boy, wow. I do. I mean, I think wilt is a really huge problem. I mean, of course, wilt is not the problem. The problem is we're not making enough progress on wilt. Yeah, I mean, cancer, despite what I was just saying, is a really, really difficult foe because it is evolutionary in its nature. whatever you throw about it, you always have this incredible power of natural selection fighting against you, and that is always going to be smarter than humans. And so, until we have a solution that is either wilt or something of the power of wilt, where you are really making the body completely incapable of becoming cancerous, we are going to have the fear stalking us. And so that is something that, if I'm kept up awake at something from uh, the whole panoply of what aging throws at us, that is it.
Yes, thank you very much, Michael, for that response and for joining us today and giving such a comprehensive presentation. I think this is the kind of presentation that our viewers will want to revisit perhaps multiple times in order to glean more details from it, more valuable information about where each of these strands of the SENS approach is at currently and the sometimes quite impressive progress that has been made during the past decade and a half. So thank you very much to Michael, to our esteemed panelists, and to our audience for a thoroughly enlightening salon. Live long and prosper.